So hi, I'm Christopher Stewart. I'm a research fellow at Newcastle University. So the gut microbiome is the collection of microorganisms which colonize the gut. And by microorganisms, we mean things like bacteria and fungi. So most of the time you can't see these, but they line the mucosal surfaces of our bodies. It's particularly important to understand the infant microbiome because we know the microbial community changes most over that first year of life, so we regard this as the sort of critical window of development. And how that microbiome changes over the first year of life and what factors are influencing that development are important to understand, not least because the microbes that colonize the gut are interacting with our own human cells that line the epithelium and also our immune system. And that interaction over the first year of life in particular can predispose an infant to both short and much longer term health and disease outcomes. So actually I started my research career as a PhD student exploring preterm infant microbiome, and I moved away from that a little bit during postdocs, but I've gone full circle now, and in my own independent group we extensively study the preterm gut microbiome, both characterizing how it develops from birth, the factors that impact the microbiome development, but we are taking that one step further now to unpick how the microbes are interacting with each other, but also the infant's own host cells. And we do this using a model system, and we can then unpick that microbial host interaction and how that potentially is involved in multiple disease processes. For my Fleming lecture, I'll be starting with the PhD work, which was very much focused on preterm infant, and in particular looking at a disease called necrotizing enterocolitis, which is an inflammatory mediated disease of the bowel, and it's also the leading cause of death in that population. So that's where it all began for me, and that's where I'm going to start the lecture. And I'll talk about some of our early evidence that suggests the microbiome probably we don't see a particular signature associated with disease onset, but on the flip side, we do see that infants who have a high abundance of a bacteria called bifidobacterium never went on to get this disease. And so that really was the start of a long journey for me, which is now where I'll end the talk, and that's talking about our most recent results, unpicking that a bit further, looking at the impacts of probiotics containing bifidobacterium and how that impacts the preterm infant gut microbiome, but also the microbe-host interaction using an organoid model system that we developed over the last few years. My entry into a research career was somewhat unexpected to me, even as I approached the latter stages of my PhD, but it was during this period that I got to do a microbial ecology project, which actually was looking at a lake sediment core and trying to understand how the microbes change through time. But then when I finished that, I found out there was a PhD opening up using similar techniques, but looking at the infant gut and how that sort of viewing that as a microbial ecology landscape and trying to understand more about that as an ecosystem. And so it was basically applying the techniques from the environmental project and applying that to a clinical project. And I got incredibly fascinated about the concept of doing that. And then meeting the clinicians involved in the project, seeing firsthand their excitement, passion, and frankly determination to improve the outcomes of their infants was incredibly inspiring for me, and ultimately what led me down this path. So as I look back on my short career to date, I can certainly recognize now, I guess, inflection points or particular moments that in retrospect were quite critical to the path that I then went down, which has led me to be here today. And certainly one that springs to mind is, as a PhD student, I was working with Suzanne Kennedy, who was helping me by sending me kits to test, and I was helping in return by testing these kits for the company. My interaction with Suzanne really just started out as email exchanges, very much focused on the science, and really began to develop into a strong collaboration. And in the end, that led to her inviting me out to America, to Baylor College of Medicine, to do a postdoc with her and Joe Petrosino. So an absolutely fantastic opportunity for me that I really don't think otherwise would have came about without that sort of serendipitous interaction with Suzanne and then all the work that then followed. Winning the 2022 Fleming Award, it means so much to me, and I know it means a lot to my team. And ultimately these awards, whilst given to an individual, are really a recognition of the many, many people who have contributed to the work. And that is from PhD supervisors to the clinical collaborators that I work day in and day out with, as well as the students and postdocs who have contributed enormously, of course, to the experiments. And also must pay particular recognition to Microbiology Society members such as Alan Walker and Lindsay Hall, who over the years have been incredibly supportive of me, have offered critical advice when I've needed it, and that for sure has played a huge part in me getting to here and accepting this award today.